DNA replication. So DNA has a double helix structure made up of two parent strands. Between each strand are nucleotide pairs that are bonded with hydrogen bonds. DNA has a sugar phosphate backbone, which you see right here. DNA strands are complementary and anti-parallel to each other. Because they are anti-parallel, they go in opposite directions called 5' and 3'. DNA replication begins with an enzyme called helicase. Helicase unzips the DNA strands so that each parent strand becomes a template of new DNA. After helicase unzips the DNA, single strand binding proteins, or SSBP, binds to the unpaired nucleotides and keeps the replication fork open for DNA replication to take place. Topoisomerase reduces supercoiling in DNA. An enzyme called primase, which you see here, makes RNA primers, which are short fragments of single-stranded RNA. They allow the DNA polymerase III to start synthesizing the new DNA strand, starting from the 3' end of the RNA primer. On the leading strand, the DNA polymerase is able to make new DNA continuously. On the lagging strand, primase also adds a primer for DNA polymerase to begin making DNA. But because DNA polymerase III only moves in a 5' to 3' direction, it can only synthesize DNA in portions known as Okazaki fragments. After DNA polymerase fills in all the sections between the primase, DNA polymerase I replaces the RNA primers with new fragments of DNA. Okay. DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. So, DNA replication is known as semi-conservative because each DNA molecule is made up of one old conserved strand of DNA and one new one.